In this lecture, we'll detail glycolysis, which is one of the central metabolic pathways. It's really common in all of life as we know it on this planet. Um, the title slide here, we're going to go through glycolysis in detail in this lecture, and we'll see there's 10 steps to it, and we'll see every step in some detail. But I like this figure on the title slide because it gives you really kind of a good overview of what's going on. You can break glycolysis down into two parts. The first part is an energy investment part where you're actually putting in energy in the form of ATP, right? And in that part, you're taking glucose, which is a 6-carbon sugar, and you're breaking it down into 2 3-carbon sugars. Okay. So that's the first part. And the second part is where you're getting an energy payout. And in that part, you're taking your 2 3-carbon sugars and converting them to 2 pyruvates. And by doing that, you're getting out 2 NADHs and 4 ATPs. Okay, so you use 2 ATPs and you're getting out 4 ATPs. So there's a net payoff to glycolysis. Just a reminder, last time we covered chapter 13. This was just general bioenergetics and reaction types. A reminder of the 6 different biochemical reaction types. They're shown here, and the name of the enzymes here that carry out these types of reactions are shown in blue. Okay, ligase, isomerase, hydrolase, lyases, which is the same name as the type of reaction. Transferases catalyze group transfer, and oxidoreductases do oxidation reductions. Okay. The one thing I wanted to point out about these reaction types and enzyme types is that the way we classify enzymes with the EC number, which stands for enzyme class, and there are 6 classes of enzymes, those correspond to the 6 reaction types. Okay, they're just in sort of a reverse order here, where the class 1 is an oxidoreductase and class 6 is ligases, and we presented it in the reverse order in the class. But just wanted to point that out because when you see an enzyme, you'll see the EC and then like certain numbers after it to tell you what type of enzyme it is. That's telling you, the first number in there is telling you the type of reaction it catalyzes. Okay. So today we're going to cover chapter 14, or at least the first part of it, and talk about glycolysis. Okay, and this is actually from a previous class. We're covering the whole 10 steps of glycolysis today. Okay, so the overview for chapter 14, there are a couple key topics. Today we're going to be talking about glycolysis, which is using glucose to harness energy. The other parts we'll talk about next time, fermentation. This is metabolism under anaerobic conditions, how we continue to do glycolysis under anaerobic conditions. We'll also talk about gluconeogenesis, which is the synthesis of glucose. You can kind of think of that as glycolysis being run in reverse. It's not quite that simple, but it's that's a decent way to think about it. Right. And then finally, there's another pathway called the pentose phosphate pathway, where you oxidize glucose to produce NADPH, and we'll briefly talk about that as well. That next time. Okay, but today we're going to be focusing on glycolysis. Okay, and glycolysis is as I mentioned, a central metabolic pathway because it's thought that this was probably one of the first energy producing pathways or group of reactions that occurred on the planet. So essentially every cell on the planet does glycolysis in the same way. From what I've been told, there are maybe a few examples of cells that have maybe like a slightly different reaction for certain steps of glycolysis, but by and large, and I don't actually know the exact cells that do that. I've just been told that there are some that kind of bypass certain steps, but you can think of as a whole that pathway, at least most of that pathway, is in every cell on the planet. Okay. So why is that? Well, glucose is an excellent fuel. It yields a good amount of energy when it's oxidized. It can be efficiently stored in a polymeric form. And we talked about those when we covered chapter 7, right? Glycogen and starch, amylose, amylopectin, are all just polymers of glucose. Right? Many organisms and tissues actually meet their energy needs on glucose alone. And the brain is one of those organs that can survive primarily, or does survive primarily, based on glycolysis and sugar, I should say, glucose. Glucose, it's also a versatile biochemical precursor, right? Some bacteria can use glucose to build carbon skeletons of basically everything they need. All the amino acids, membrane lipids, nucleotides, and cofactors needed for metabolism. Okay, so it's a very useful molecule. Okay. There are 4 major pathways of glucose utilization: okay. Storage, glycolysis, pentose phosphate pathway, and the synthesis of structural polysaccharides. Okay, so storage, glucose is stored in the polymeric form, and again, those are things like starch or glycogen, depending on whether you're a plant or an animal. Okay, and that's done when there's excess energy. Right? Glycolysis is the opposite of that, sort of. It's the breakdown of glucose to generate energy via its oxidation. Okay, and that meets short-term energy needs. Okay, the pentose phosphate pathway generates NADPH via the oxidation of glucose. So it's similar to glycolysis in that you're oxidizing glucose, but instead of getting energy out of that, you're getting a cofactor, a reduced cofactor NADPH out of that. And then that NADPH is then used for other cellular processes, such as detoxification or the synthesis of lipids and nucleotides. Okay. And then finally, the synthesis of structural polysaccharides. These are things like cell walls of bacteria, fungi, and plants. So this is primarily a pathways that in bacteria, fungi, or plants, and not in animals. Okay, so we really won't talk a whole lot about that in this class. Okay, and this is just a figure showing that same kind of information that was on the previous slide. Probably a little bit easier to kind of understand that in graphical form. Okay. So why is glycolysis important? Well, glycolysis itself is enzyme catalyzed reactions that convert glucose into a molecule known as pyruvate. Okay. It turns out pyruvate can be oxidized further aerobically, and it's also can be used as a precursor in biosynthesis. So pyruvate's a very important molecule, and as we'll see in chapter 16, pyruvate, when it is broken down further in the citric acid cycle, it generates a whole lot of ATP. So it's a very, very high energy molecule, or potential to make a lot of cellular energy. So you need to make pyruvate, and one of the ways to do that is by converting glucose into pyruvate. So that's one reason why glycolysis is so important. Right. some of the oxidation-free energy if glycolysis is captured by the synthesis of ATP and NADH. Okay, ATP is, that can be used directly as cellular energy. NADH, that's a reduced cofactor, and it can then transfer its electrons to the electron transport chain in the mitochondria, thus generating even more ATP. And we'll talk about that in chapter 19. Okay, so glycolysis can produce energy itself, and it can produce pyruvate, which then goes to produce more energy or other biomolecules. Okay. Research of glycolysis played a really big role in the development of biochemistry. This was, since it is such a central pathway in very simple organisms have glycolysis, organisms that are easy to study, it was really one of the first pathways, metabolic pathways, that was studied. And by studying this, scientists got an understanding of the role of coenzymes in catalysis. They also discovered the pivotal role of ATP, how it plays in cellular energy, the development of methods for enzyme purification. So a lot of these enzymes involved in glycolysis were some of the first enzymes that were purified, and it also inspired the next generations of biochemists. So as I mentioned before, glycolysis was probably one of the earliest energy-producing pathways. It developed before photosynthesis, when the atmosphere of on the Earth was still anaerobic. So glycolysis doesn't require oxygen around, or oxygen atmosphere to go. So anaerobic organisms still can use glycolysis, unlike the main respiratory chain, electron transport chain in the mitochondria, that requires oxygen. Glycolysis doesn't. Okay. So early organisms on the planet, their task was to extract free energy from glucose anaerobically. Okay, and the solution was you first activate it by phosphorylation, you add a phosphate to it, and then second, you collect the energy from the high energy metabolites. And so those are really kind of the two parts of glycolysis, maybe like a preview of the two parts of glycolysis. Okay, so this is the figure that the book uses for the whole pathway of glycolysis. As you can see, you start with glucose, and at the end you get two pyruvate molecules out of it. And I like this figure because it shows the actual molecule, the structures of all the molecules in each step. So that's pretty good. Pretty good information there. And it shows in blue the enzyme name for each step. So we show a lot of different figures in this class on glycolysis. And I think there's not really one figure that is the best. I think it's good to look at, the different figures, because you kind of can get different information out of them. So this one really, I think the benefit of it is showing that the actual molecule structure for each step, it's probably the best, its best feature, as well as is highlighting the enzyme names in blue. Okay, we're going to look a little bit more closely at all of these steps, so you don't need to memorize this right now. The first 4 to 5 steps are considered the preparatory phase. And I say 4 or 5 because it depends on the figure you're looking at. Some figures just call the first 4 steps the preparatory phase. Others include the 5th step here. Okay, so it I'll probably have to, on an exam, maybe stay consistent, since this is figures from our book. We'll just call it the first 5 steps, I guess. The payoff phase is the next step 6 through 10, and that's where you get energy out. So the first 5 or 4 you're putting in energy, and the last 5, 6 through 10 you're getting out energy. So let's look at the first part, the preparatory phase. Okay, and there are 4 main reactions here that we'll look at in detail. And then we'll kind of consider the 5th step with the next phase at the beginning of the payoff, or in between the preparatory and the payoff phase. Right, and you can kind of think of this as you're investing money into something that you're hoping is going to pay off in the future. So it's an investment. Okay, and then we'll go through the payoff phase after that. Right, this figure again, it's from the same book, but now it's having the 5th step here is kind of not in the preparatory phase or the payoff phase. We'll just kind of lump that 5th step in the preparatory phase. Okay, and the payoff phase will be reactions 6 through 10, and this is where your investment gains interest and starts to pay off. Here's yet another figure for glycolysis. This one I won't go a lot through, but this is what the book describes as the chemical logic of glycolysis. And this one is helpful because it shows you the numbers of carbons, right? So we have a 6-carbon sugar, glucose, and then it's showing you at those carbons, is there a carbonyl group or an alcohol, and it's showing you kind of what's going on at those different carbons. So this one might be helpful for some of you to read through it, maybe help you understand it a little bit better, or maybe you're able to remember some of these steps a little bit better by thinking about the chemical logic of it. Okay, so let's start with the first step. Okay, the first step is the reaction of glucose going to glucose 6-phosphate, and that's catalyzed by an enzyme called hexokinase. Right? Hexo meaning it's 6, in this case a 6-carbon sugar, and kinase tells you that it's an enzyme that catalyzes the transfer of a phosphate group. Okay, so kinases transfer phosphate groups, and hexo is indicating we're doing this to a 6-carbon sugar. Okay, so that name tells you what that enzyme's doing. So if you think about it, if you remember just the name of the enzyme, you'd be able to remember, okay, what's happening here? Glucose is getting a phosphate added to it. Okay. And it's doing that at carbon 6. Okay, so it's carbon 6, the alcohol group on that carbon is being replaced by a phosphate. Okay, this is also it's using ATP. So right off the bat, first step in glycolysis, we're using energy, which it seems kind of counterintuitive, right? You're supposed to be getting energy out of glycolysis. In the very first step, we're actually using energy, but there are good reasons for that. Okay. The delta G naught prime of this reaction is negative, a fairly high negative value, and that means it's going to be very favorable to go forward and in a sense irreversible. You can also think of this as a priming step, like you're priming a pump. You have to add some energy to it, right, before the pump starts working. So what reaction type is this? If we think about the reaction types we talked about in chapter 13, it's a group transfer. You're transferring a phosphate. Okay. And the reaction itself, if you just look at the reaction, glucose plus inorganic phosphate going to glucose 6-phosphate, that has a very positive delta G value, which would be unfavorable, but you're coupling it to the hydrolysis of ATP, which is a very negative delta G. And so this reaction, by coupling those, becomes favorable. Okay, the rationale behind adding a phosphate to glucose in this step is that the glucose by itself can very freely go in and out of cells. When you add a phosphate group to it, what it does is it traps that glucose inside the cell. Right? It also, you're changing the molecule from glucose to glucose 6-phosphate. So in a sense, you're lowering the concentration of glucose inside the cell, which allows further uptake of glucose. Okay, so that would be the rationale behind it. Okay, the process, again, uses energy in the form of ATP. Okay, you can think of this as priming, a priming step. Okay. The enzyme is called hexokinase, at least in eukaryotes. You have a nucleophilic oxygen at carbon 6 of glucose. It attacks the gamma phosphate of ATP. ATP has a magnesium ion bound that facilitates this process by shielding the negative charges on the ATP. Okay. And this is highly thermodynamically favorable, and which makes it an irreversible step. Okay. It's regulated mainly by substrate inhibition. And we'll talk about the regulation of glycolysis in a later lecture, so don't feel like you have to understand that quite fully yet. Okay. Moving on to step 2. Step 2 is a isomerization step where we have glucose 6-phosphate, which was the product from step 1. That's what we're starting with in step 2. That is being converted into fructose 6-phosphate. Okay, and you can abbreviate. In this class, it's fine if you abbreviate glucose 6-phosphate as G6P and fructose 6-phosphate as F6P, if it's easier. This reaction is catalyzed by an enzyme called Phosphohexose Isomerase. Right? Phosphohexose is the sugar, 6-carbon sugar with a phosphate, and isomerase is telling you what it's doing. It's catalyzing an isomerase reaction. So this reaction type is an isomerization. You're changing the glucose, which is an aldose, into fructose, which is a ketose. The delta G on it is delta G naught prime is 1.7 kilojoules per mole. So that's pretty very close to zero. Slightly unfavorable, but it's close to zero. Okay, so the rationale behind this step is that the carbon 1 on fructose is going to be easier to phosphorylate by in the next step, the enzyme in the next step, which is PFK. Okay, so we're getting it ready for phosphorylation in the next step. Okay. It also allows for symmetrical cleavage by the enzyme of the 4th step. Okay, so this step 2, you're really getting this substrate ready for the next couple steps of glycolysis. As I mentioned, glucose is an aldose. They can isomerize into ketoses via an ene-diol intermediate, which we'll see here in a second. Okay, the isomerization is catalyzed by an active site glutamate, glutamic acid residue, via general acid-base catalysis. Okay, and again, we'll see that in the mechanism. This is slightly thermodynamically unfavorable, but it is because the delta G naught prime is close to zero, it's considered reversible. So how do you get this reaction to go forward? The product concentration is kept low to drive the reaction forward. And you can think of that as pulling the reaction forward. Okay, once the product of this reaction is made, it's used in a next step, which keeps that concentration low, which drives the reaction forward. You're pulling it forward. So there's two ways you can get around having a slightly unfavorable delta G naught prime. One is you can, and this is basically Chatelier's principle, you can overload the reactant concentration, keep the reactant's concentration high, which will push the reaction forward, or you can take away the product and keep that concentration really low, and that's known as pulling the reaction forward. In glycolysis, many of these reactions, the products are kept really low, which pulls it forward. So that'll be a common theme that we'll see more. Okay. If we look a little bit closer at this mechanism, okay, this is the mechanism of Phosphohexose Isomerase and its general acid-base catalysis. And before we get too involved in this, I'll be showing the mechanisms of a few of these enzymes in glycolysis. But please note that I wouldn't expect you to be able to draw these from scratch on an exam. This isn't really organic chemistry, so I wouldn't expect that. I think it's helpful in understanding what's going on, and it maybe will help you remember what the product of a certain step is by remembering these or learning about these mechanisms. But I wouldn't expect you to draw them from scratch. I might show you a picture of a step of a mechanism and ask you, okay, what enzyme is this? So then I'd expect you to be able to look and be able to say, okay, what substrate is in that active site? Okay, what kind of mechanism is occurring? What residue on the enzyme is being used in the active site? Using all that information to be able to come up with the right enzyme. Right? But again, you wouldn't have to draw these mechanisms from scratch. On a paper exam, I might ask you, take away the arrows and have you draw the arrows in. I think that, might be something you could do, but probably not going to do that on an online exam. Okay, so this is Phosphohexose Isomerase. Its substrate is glucose 6-phosphate. Okay, the first step, glucose 6-phosphate, binds in the active site, and the ring opens. Okay, and we see that here. Okay, and it gets close to a residue. In this case, it's a glutamate residue that acts as a base in that it's going to abstract the proton at carbon 2 of glucose 6-phosphate. Okay, so it abstracts that proton. The electrons then push around. You get this double bond formation between carbon 1 and carbon 2. Oxygen on carbon 1 picks up a proton to form a alcohol, and this is that ene diol, ene for the double bond, and diol because it has two alcohols on it, intermediate. Okay, so that is, the first step is the formation of the ene diol intermediate, and then the second step is that you have a proton. Now on the proton on the alcohol on carbon 2 is removed, which makes the carbonyl on carbon 2 instead of on carbon 1. Okay, so that's how that carbonyl kind of flip-flops the position from carbon 1 to carbon 2. Okay, step 3, this is our second priming step, and we're again using ATP. So our starting reactant would be fructose 6-phosphate. Okay, we're adding an ATP, a phosphate from ATP on carbon 1 now, and so we're phosphorylating carbon 1, and that product we make would be fructose 1,6 bisphosphate, or F16BP. Okay, and that's another thing I should mention. The bis is telling you that there are two phosphate groups there. And this enzyme that catalyzes this reaction is called phosphofructokinase 1, or PFK1. Phosphofructo because that's your substrate, and kinase because you're, again, adding a phosphate group. And this reaction type, again, is a group transfer. The rationale behind this step, it's a further activation of glucose. You're adding another phosphate, so you're activating it. You're putting some more potential energy in it. Okay, and it allows for one phosphate on each 3-carbon sugar that's going to be produced after step 4. Okay, so you're making it so that you can cut this 6-carbon sugar in half, and each 3-carbon sugar that's left is going to have a phosphate. Okay. This is the first committed step of glycolysis. So the first 2 steps, the first step, you're just, in a sense, you're adding a phosphate to glucose to keep it inside the cell. The second step is an isomerization to change glucose 6-phosphate into fructose 6-phosphate, but that's reversible. Okay, and then there's other pathways that break off from there. But this step, after this third step, you're committed to glycolysis. So fructose 1,6-bisphosphate is committed to become pyruvate and yield energy. This process uses energy again, ATP. This is the second priming step. Highly thermodynamically favorable, which makes it irreversible, as you'd expect, because you're coupling this to the hydrolysis of ATP. PFK, and this is phosphofructokinase 1, but a lot of times we just abbreviate it as just PFK. It's a highly regulated enzyme. And why is that? Well, this is the first, after you're done with this, you're committed to glycolysis. So you don't want to make fructose 1,6-bisphosphate when you have a lot of ATP already around. You have enough ATP, there's no reason to burn glucose. Okay, so this is going to be a very highly regulated step. Okay, and what it's regulated by, one is ATP. That's pretty obvious. It's also regulated by something called fructose 2,6-bisphosphate, right? It makes fructose 1,6-bisphosphate, but a molecule that's very similar, fructose 2,6-bisphosphate, is one of the molecules that regulates it. And again, we'll talk about regulation in more detail later. So don't feel like you have to understand that yet. Okay, step 4 is an aldol cleavage of fructose 1,6-bisphosphate. Okay, and it's done by an enzyme called aldolase. Okay, you can think of aldols. I don't know if this is this is one of those enzymes that you kind of just have to remember. At least in my head it is. There's not a whole lot of, it doesn't make a whole lot of sense, that the name of this to me anyways. So it's one that I just kind of have to remember. Okay. So the reaction, it catalyzes fructose 1,6-bisphosphate, or F16BP. It cuts it in half, and you're going to get 2 3-carbon sugars. One of those is glycerol aldehyde 3-phosphate, which is abbreviated as GAP or GAP. Okay, and that is shown here. It's an aldehyde, an aldose, and there's 3 carbons, and it's phosphorylated at that 3rd carbon. Okay. The other one that it produces is called Dihydroxyacetone Phosphate. And that is a ketose, okay, 3-carbon sugar. The carbonyl is a ketone, not an aldehyde. Okay, and it's phosphorylated at carbon 1 in this case. Okay, so another helpful thing on this slide is it's numbering the carbons according to the numbering scheme in the original glucose. So the dihydroxyacetone phosphate, or DHAP, comes from the first three carbons of glucose. The GAP, glycerol aldehyde 3 phosphate, comes from the carbons 4, 5, and 6. Okay, this reaction has a very high positive delta G. Okay, and what type of reaction is this? It's an lyase. Okay, so lyases are enzymes that the reaction, it's a little bit kind of tricky to define, right? And so this is one of those. Okay, the rationale of this step is that the cleavage of a 6-carbon sugar into two 3-carbon sugars. Okay. High energy phosphate sugars are 3-carbon sugars, so you're creating very high energy intermediate sugars that can then be used to phosphorylate ATP in further future steps. The reverse process is known as an aldol condensation, which is why that enzyme is called aldolase. That doesn't really help me remember it, but maybe it'll help you remember that. I'm just not. I don't think as like an organic chemist would, and think about the reverse reaction here. That's why it's named that. As a bit of an aside, some of these enzymes are named seemingly more for their reverse reaction, and that's because when they were discovered, when the people that discovered the enzymes were looking at them in a test tube, the conditions were such that the reaction was actually going in reverse. Okay, so a lot of enzymes are named kind of for the reverse reaction, the reverse of the biological reaction. Okay, animal and plant aldolases employ what's known as covalent catalysis, which we'll see in the mechanism when we look a little bit closer. Okay, and as I mentioned, it's thermodynamically unfavorable, and it actually, even though it has a very large delta G, it is reversible. Okay, and the reason and the way this goes forward is that the GAP glyceraldehyde 3 phosphate concentration is kept really low, which pulls this reaction forward. Okay, and again, that's a kind of a common theme here in glycolysis. Okay, so if we look at the mechanism, and this one, it's one of the more complicated mechanisms. Okay, and this one I definitely wouldn't expect you to draw from scratch, but I'll just point out some of the things that are unique about it. Okay, one thing that's unique is that it's using a lysine in its active site. Okay, that lysine is going to attack carbon 2 on fructose 1,6 bisphosphate. And it does that, and it forms this tetrahedral intermediate that's covalently bound to the enzyme. Okay, this intermediate then it undergoes a rearrangement. There's a base abstraction of a nitrogen from the lysine, okay, and you have the oxygen at carbon 2 here. It's going to leave in the form of water, and then you get this shift base intermediate that's formed. Okay, when that's formed, you then get another proton abstraction, this time at carbon 4, the oxygen on carbon 4, and that's what cleaves the carbon-carbon bond. Okay, that then releases, after that step, releases the first product, the glycerol aldehyde 3 phosphate is released. Okay, and that's again carbon 4, 5, and 6 from the original glucose. Okay, and then you have an isomerization step where you have a double bond that was formed the carbon-carbon double bond that rearranges again into a Schiff base. And then you have the Schiff base is hydrolyzed, and the dihydroxyacetone phosphate leaves as the second product of this reaction. Okay, so this is the overview of that reaction. And I should mention too, you can find animations of these mechanisms, which might help understanding them a little bit better online. I had some links in here to some that I had, but they were all, they all use Flash Player, so they're kind of useless now. Okay, step 5, and we can think of this as, we'll just call this in our class, step 5 is the last step of the preparatory phase. Right? And what you do in step 5 is you convert your DHAP, the one molecule of DHAP, into another molecule of GAP. And this reaction is catalyzed by an enzyme called triose phosphate isomerase. Okay, triose means there's a 3-carbon sugar. Triose phosphate is a 3-carbon sugar with a phosphate. And isomerase is telling you what it's doing. It's isomerizing that. Okay, so the reaction type is an isomerization. Okay, so you're taking dihydroxyacetone phosphate and you're converting it into glycerol aldehyde 3-phosphate. The delta G naught prime is a positive value, not really a very high, but it is positive. Okay. The rationale behind this step is that it's going to allow glycolysis to proceed by one pathway. So if you didn't do this step and you had one dihydroxyacetone phosphate, one glycerol aldehyde 3-phosphate, those would have to go through 2 separate pathways after that. But now you convert it into 2 molecules of GAP, and you can send them both down the same pathway. Okay, so in a sense, it simplifies things. Right. Aldolase creates, as we mentioned in that previous step, step 4, you get a DHAP and a GAP, and only GAP is the substrate for the next enzyme, so you need to convert your DHAP into GAP. Okay. This would, after this step, that completes the preparatory phase. Again, this is thermodynamically unfavorable, and it is a reversible step. How it's driven forward, same rationale we used on step 4, GAP concentration is kept low to pull this reaction forward. Right, so this is another figure I think that's actually pretty good at summarizing the preparatory phase of glycolysis in terms of where each carbon ends up. Okay. So this is the fructose 1,6-bisphosphate and carbons 1 through 6 as they would be numbered in glucose. Okay. After aldolase, right, carbon 1, 2, 3 is on the DHAP, and carbon 4, 5, 6 is on glycerol aldehyde 3-phosphate. Okay, but then this DHAP is going to be converted into a GAP. So where do those carbons go? And so carbon 1, you can see that's phosphorylated. So carbon 1 is going to be like the carbon 6 on glycerol aldehyde 3-phosphate. Right? And so that is summarized here. So from our glycerol aldehyde 3-phosphate, we can now call these carbons 1, 2, and 3 of glycerol aldehyde 3-phosphate. Okay, where did they come from? Carbon 1 on GAP came from either carbon 4 or carbon 3 of glucose. Carbon 2 on GAP came from carbon 5 or carbon 2 on glucose. And carbon 3 on GAP comes from carbon 6 or carbon 1 on glucose. Okay, why this is important, that's one of the favorite types of questions in this part of biochemistry, is to say you have glucose that's labeled with a radioactive carbon 14 at position X. What position in this product molecule GAP or pyruvate, what position of that is going to be radioactive. And so this helps you kind of follow it through. Okay, so that was the preparatory phase. Moving on to the payoff phase. All right, step 6 is the first step of the payoff phase, and it's the first step that actually yields energy. And I denote that here with the little $sign. Okay. Okay, step 6, what's going on? You're having glycerol aldehyde 3 phosphate. I should mention now the stoichiometry becomes 2. It becomes doubled because 2 glycerol aldehyde 3 phosphates are produced for each glucose. But that's not going to be shown here on these slides. But just kind of remember that in the back of your mind. Right. So in step 6, you have GAP, and you're adding an inorganic phosphate to it. Okay. And you're producing 1,3-bisphosphoglycerate, or 1,3-BPG. Okay. By doing this, you're also taking an NAD+, an oxidized nicotinamide adenine dinucleotide, and you're reducing it to NADH. This is all catalyzed by an enzyme called glycerol aldehyde 3 phosphate dehydrogenase. Okay, so that enzyme name dehydrogenase is telling you that this is an oxidation reduction reaction. So you're oxidizing glycerol aldehyde 3 phosphate by adding this phosphate group to it. In a sense, you're adding another oxygen there. But you're reducing, by doing that, you're also reducing NAD+ to NADH. Okay, and so it's not very obvious yet, but this NADH, the reduced cofactor, has extra electrons in it, and it can then transfer those electrons to the main respiratory chain, and that's going to generate ATP. Okay, so that's where you're getting a payoff, okay, this reduced cofactor. And remember, for every molecule of glucose, you have 2 of these reactions occurring, so you're going to be making 2 NADHs. The delta G naught prime of this is, again, a positive value. Okay. The rationale behind this, generation of a high energy phosphate compound, so you're adding another phosphate to it to make it a high energy compound. So you're building a high energy compound as your product. Okay. It incorporates the inorganic phosphate, which then allows for a net production of ATP via glycolysis. Okay, so you're adding another phosphate to your 3-carbon sugar, which then can be transferred to ADP to make an ATP. Okay, so by doing this, so let's just take a step back. In the preparatory phase, you've added phosphates to glucose. You added 2 of them. If you didn't do this step and you just transferred those 2 phosphates to ADP in the next payoff phase, you'd only be making 2 ATPs. So you'd be breaking even on ATPs. So this addition of an inorganic phosphate to this molecule, and you're doing it twice, it's extending the amount of ATP you can make, which actually makes it so you can make a profit of ATP. All right, this is the first energy yielding step in glycolysis. And again, that's because you're making the NADH. You're oxidizing an aldehyde with NAD+ to give reduced NADH. This enzyme uses an active site cysteine, which is unique, at least unique in glycolysis. It forms high energy thioester intermediate, which that's subject to inactivation by oxidative stress. We really probably won't talk about that much, but this enzyme is susceptible to, if you have a lot of oxidative stress going on, that is susceptible to oxidative stress. Thermodynamically, again, it's unfavorable, and this is a reversible step. Okay, and why, I should have mentioned this earlier, but why it's important to understand if these are reversible or not. When we talk about gluconeogenesis, the reversible steps can just be run in reverse if you need to produce glucose. Um, the ones that are not reversible, those steps you need to have a bypass for. So that those will come up again. Okay. How this reaction goes forward in glycolysis, it's coupled to the next reaction to pull it forward. So step 6 and step 7 are coupled together. Okay, the mechanism of glycerol aldehyde 3-phosphate dehydrogenase is shown here. And again, it's unique in glycolysis in that it has a sulfur at the active site. It also has an NAD plus cofactor. Okay, glycerolaldehyde 3-phosphate will bind there. Okay, you're going to get a nucleophilic attack by that sulfhydryl, or I guess it's not a sulfhydryl anymore because the hydrogen's gone. But that sulfur is going to attack the carbonyl of glycerolaldehyde 3-phosphate, and you produce this covalent intermediate, and it's a thioester intermediate. Right. The proton and the electrons from that bond are then transferred to NAD to get NADH, and you produce intermediate, oxidized intermediate. Okay. Then the inorganic phosphate comes in. You have NADH leaving, an NAD plus coming in, and an inorganic phosphate. That inorganic phosphate then does nucleophilic attack on the carbonyl carbon, and you have the 1,3-bisphosphoglycerate product that leaves, and you reset your enzyme. Okay, and here's the steps 1 through 5 just shown together. Okay, step 7. This is the first production of ATP in glycolysis. Okay, so this is our second energy producing step, and it's the first one that produces ATP. Okay, so in this step you have 1,3-BPG and ADP, and you produce 3-phosphoglycerate and ATP. So you're transferring the phosphate group from 1,3-BPG to ADP. And this is catalyzed by a reaction known as phosphoglycerate kinase. So phosphoglycerate is the substrate, and kinase is the type of enzyme. And again, this is a group transfer. That kind of cut off by the piggy bank there, but it's a group transfer reaction. 1,3-BPG is a very high energy molecule, and transferring that, breaking that phosphate bond there to it releases a lot of energy. And so it's even higher energy than ATP, because this delta G value here is very negative. Okay, this is a substrate level phosphorylation to make ATP, right? And so that's that is important. Okay, and again, 1,3-BPG is a very high energy compound. It's one of the few compounds that's higher in energy than ATP. So it's one of the few compounds you can use to donate a phosphate group to ADP to make ATP. Okay, again, this is very favorable. It is, however, reversible, which is a little bit counterintuitive, being that it is such a negative delta G value. But the reason it's reversible is because it's coupled to the GAP-DH reaction, step 6. So because it's coupled to step 6, it makes this reversible. Okay, step 8. Step 8 is the migration of a phosphate. So we have 3-phosphoglycerate, 3-PG, and that phosphate group you can think of just moves to from carbon 3 to carbon 2. That's not actually that simple, as we'll see in the mechanism, but you can kind of think of it that way. And you produce 2-phosphoglycerate. And this enzyme that catalyzes this is phosphoglycerate mutase, or PGM. Right. This is an isomerization reaction. Although it's sort of more of a group transfer when you look at the mechanism, but you can think of it as an isomerization because of the net result of the going from the reactant to the product. Okay. The rationale behind this, why you're moving the phosphate from carbon 3 to carbon 2, you're trying to form a high energy phosphate compound, and it turns out that the phosphate on carbon 2 is going to give you that is going to allow you to form a higher energy compound. Okay, this is an apparent migration of groups. And enzymes known as mutases catalyze apparent migration of groups. It's not, as it turns out, it's not that actual phosphate group moving. It's going to be a different one. Okay, so in this active site there are two histidines, and one of the active site histidines is post-translationally modified with a phosphate group. Okay, so there's a phosphate group on a histidine in there already. Okay, that phosphate group is going to be attached to carbon 2, and then the phosphate that was on carbon 3 in this substrate is going to move to the other histidine. Okay, this produces 2,3-bisphosphoglycerate intermediate. We'll see that in the mechanism. The phosphate that was on the substrate ends up bound to the enzyme at the end of the reaction. So again, it's not the same phosphate moving, but that's why this is a mutase, not an isomerase enzyme. Okay. This is thermodynamically unfavorable and reversible. Okay, and this is a rare case where the reactant concentration is kept high to push the reaction forward. Right, so here's our mechanism. Right, we have two active site histidines. One has a phosphate group. The phosphate group on that active site histidine is going to be placed on carbon 2. That gives you a 2,3-BPG intermediate. All right, and that 2,3-BPG intermediate can then give up the phosphate on carbon 3 to the other histidine. Actually, it would be back to the same histidine that had the phosphate group to begin with. Okay, and that resets the enzyme, and that gives you your product, 2-phosphoglycerate. Okay. Step 9. So step 9, you have 2-phosphoglycerate, and you're producing a molecule known as phosphoenol pyruvate, or PEP, or PEP. Okay, so you kind of hear that PEP or PEP being the abbreviation for phosphoenol pyruvate. Okay, and this reaction is catalyzed by an enzyme known as enolase. And I think you can remember that enol is you're making phosphoenol pyruvate. Okay, and an enol is you have a carbon-carbon double bond and usually an alcohol there, but in this case there's a phosphate group instead of an alcohol. So it's phosphoenol pyruvate. And pyruvate is a 3-carbon molecule. Okay, this reaction type, it looks like a condensation, right? Because or a dehydration, because you're removing water there. However, it's classified as a lyase. Okay, the rationale behind this is you need to generate a high energy phosphate compound, which then can be used to donate that phosphate to make an ATP. Okay, so this is our last step of making a high energy intermediate. Okay. 2-phosphoglycerate is not a good enough phosphate donor. Okay, you have two negative charges in 2PG that are fairly close, which does make it high energy, but the loss of the phosphate group from 2PG would give it a secondary alcohol, with no further stabilization. Right, so here you have a negative, just go back to the picture of the molecule. You have a negative charge here and a negative charge on the phosphate. So they're pretty close together, those negative charges. But if you lose this phosphate group, you would just be left with an alcohol here and here, which wouldn't give you any further stabilization. Okay. And so that's why you create the phosphoenol pyruvate here. And we'll explain how this leads to stabilization in a second. Okay. This reaction is slightly unfavorable and reversible. Product concentrations here, again, are kept low to pull it forward. So PEP concentrations are low, and that pulls this reaction forward. Okay. The final step of glycolysis, step 10, this is our second production of ATP, and it's actually the third energy-producing step. We have phosphoenol pyruvate plus ADP going to pyruvate and ATP. So we're transferring that phosphate group from PEP to ADP to make an ATP. Okay, and this reaction is catalyzed by the enzyme pyruvate kinase. Right? And that's that again it's an example of a enzyme name that might be probably was named for the reverse reaction, right? Pyruvate kinase suggests you're adding a phosphate group to pyruvate. Well, but the biological function of this enzyme is actually the reverse of that. Okay. The delta G of this reaction is very negative, so it's strongly favors the forward reaction, the production of pyruvate and ATP. And this again is a group transfer reaction. Okay. The rationale behind this, again, it's the second substrate level phosphorylation to make ATP. And this is actually the payoff step of ATP, because you're making, if you think about, you have two molecules of PEP that come through here for each molecule of glucose. You've made two ATPs in step, what was that, step 7, and that just breaks, you break even on that step, and this is the actual payoff in terms of ATP production. Okay, and it allows the net production of 2 ATPs per glucose. The loss of phosphate from PEP yields an enol that can, what's known as tautomerize into a ketone, and that provides another level of stabilization. And it actually also, if you want to think of it this way, it lowers the concentration of the reactant, right? When that tautomerizes, in a sense, you're lowering the concentration of the product, and you're pulling that reaction forward. Okay, again, this is highly favorable, which makes it irreversible. So this is one of the steps that needs to be bypassed when we do gluconeogenesis. This is also a regulated step. It's regulated by ATP concentration, divalent metals, and some other metabolites that we'll talk more about when we talk about regulation. Okay, and tautomerization is kind of shown here, right? This is the enol form of pyruvate. So you lose that phosphate group. You have the enol from the phosphoenol pyruvate. You have the enol form of pyruvate now that can shift into the keto form. Okay, and when it does that, you're, in a sense, this is the product of that step, the last step. And so when it tautomerizes, you're, in a sense, lowering the concentration of that product, which then allows the reaction to be pulled forward. Okay, so a summary of glycolysis. In glycolysis, what do you use? So you use one glucose. You have to use two ATPs in the preparatory phase, and you have to provide two NAD pluses for, what was that, step 6. Okay. What do you make from glycolysis? Well, you make a total of 2 pyruvates. Pyruvate can have different fates, okay, at the end. You make a total of 4 ATPs, and you also make a total of 2 NADHs. Okay. The important part about the NADHs is you have to reoxidize it to NAD plus for glycolysis to continue. Okay, and so that's when we talk about fermentation. That's the driving force of fermentation right there. Oh, sorry. So the net reaction for glycolysis is 1 molecule of glucose plus 2 NAD pluses plus 2 ADPs plus 2 inorganic phosphates go to 2 pyruvate plus 2 NADH plus 2 protons plus 2 ATPs. Okay, so you're making, in terms of energy, directly from glycolysis, you're making 2 ATPs. You're also making 2 NADHs, which then can be used to make even more ATP in the electron transport chain. Okay. The book kind of goes into the fact that glycolysis is occurring at elevated rates in tumor cells. Okay, and this is a little bit FYI because I'm not going to expect you to know this for an exam, but a lot of you might be interested in cancer biology. So glycolysis, because it's elevated in tumor cells, the enzymes, certain enzymes of glycolysis are targets for anti-cancer drugs, and some of those are shown here. Another thing about tumor cells is that you have, in general, in tumors, you have very hypoxic environments, so very low oxygen. So they can't, they don't have the same aerobic respiration going on. They're not making a ton of ATP from pyruvate. So that really drives glycolysis even further because they need to make all of their energy from glycolysis. So that really makes the enzymes of glycolysis good targets for anti-cancer drugs. Okay. So fates of pyruvate, and we'll be talking about these in the next lecture and also in chapter 16. And 19, really. So glucose going through glycolysis gives you 2 molecules of pyruvate. If you have anaerobic conditions, then that pyruvate can go to make either ethanol or lactic acid, depending on whether you're a yeast or something like a muscle cell in an animal. Okay, and those processes, the process that produces those under anaerobic conditions, that's known as fermentation. We'll be talking about that next lecture. Under aerobic conditions, pyruvate then goes to form Acetyl Coa, and Acetyl Coa is then fed into the citric acid cycle. Okay. After the citric acid cycle, it becomes 4 molecules of carbon dioxide and 4 waters. Okay, and I should mention, going to pyruvate to acetyl CoA, you're losing carbon in the form of carbon dioxide as well. Okay, so the carbons are all accounted for. Okay, and so we'll talk about the pyruvate to acetyl CoA. That's an enzyme known as pyruvate dehydrogenase complex. Okay, that, and then acetyl CoA in the citric acid cycle, that's all covered in chapter 16. Okay. There are some feeder pathways for glucose. Glucose molecules are cleaved from glycogen and starch by a molecule known as glycogen phosphorylase. Okay, when that cleaves glucose from glycogen, that yields glucose 1 phosphate. Okay, that's a little bit different than glucose 6 phosphate. That's the product of the first step, so that needs to be rearranged. Disaccharides are also feeder pathways for glycolysis. Okay, disaccharides can be hydrolyzed, and you get out the two sugars. Okay, so lactose is a disaccharide, and it's made up of one molecule of glucose and one molecule of galactose. So the glucose molecule can go straight into glycolysis. The galactose then needs to be altered or go in at a slightly different point. Okay, and the same for sucrose. Sucrose is one molecule of glucose and one molecule of fructose. Okay, again, glucose goes straight into glycolysis. Fructose goes in at a different point. Okay, so fructose, galactose, and mannose enter glycolysis all at different points. You don't need to know these. In this class, we're not going to be testing on this. In that the feeder pathways are shown here. So some different sugars you can have, right? Sucrose, enzyme called sucrase hydrolyzes that into a glucose, one molecule of glucose, one molecule of fructose. Lactose has one molecule of glucose and one molecule of galactose. That galactose then has, quite a few other steps it has to go through to actually get into glycolysis, which would be right here. This is kind of what we'd think of as glycolysis. The important thing here, what I wanted to mention that people will probably find interesting, the reason why fructose is considered really kind of bad for you is that if you look on this pathway, fructose and things like high fructose corn syrup, when you eat them, you're getting a lot of fructose in sugar, and you might think, well, why is that any different than eating a lot of glucose? Well, fructose enters glycolysis at this step. So what is that? Step 3. Okay, it goes into fructose 6-phosphate. And so fructose 6-phosphate going to fructose 1,6-bisphosphate, that isn't a very regulated step, right? That's you're kind of just putting the sugar into glycolysis and having it go straight through to produce ATP, whether you need that ATP or not. Okay. And then the back end of that, when you produce your pyruvate and acetyl Coa, when your body has a lot of that around and it doesn't need it, those get converted back into fats for storage. And so that ultimately is why the fructose is kind of like a bad quote unquote sugar. Right, so next time we're going to finish chapter 14. Primarily we'll be talking about fermentation, okay? We'll also cover gluconeogenesis and a little bit about the pentose phosphate pathway.